We meet again in Madam Nur Shamsia's biology video. Today's video is a continuation from Chapter 9 Biology Form 5 KSSM Ecosystem 9.2 Population Ecology. And today we are going to concentrate on Quadrat sampling technique. The content standard for today's lesson is 9.2 Population Ecology. The learning standard is 9.2.2 Analyze data to estimate population size of organisms using Quadrat sampling technique. There are two important data when we want to study population ecology. That are population size and population density. So we are going to look at these terms. Okay, population size is the number of organisms present in a population, whereas population density, is a number of individuals of a species per unit area of a habitat. When the area is too large, the population can be estimated by a random sampling technique. Sampling technique can be used to estimate population size. There are actually two sampling techniques that can be used. The first one is Quadrat sampling technique. The second one is mark release recapture technique. And today we are going to concentrate on the Quadrat sampling technique to estimate the population size. And this Quadrat sampling technique can be used to estimate the population size of land plants or animals which are inactive. And this technique is using a quadrat. Right, just now I mentioned quadrat. So what is quadrat? Okay, you can see the picture here. This is a sample of quadrat. So quadrat is a square frame structure which you can make out of wood, iron or plastic. And a quadrat normally we can subdivide it into few parts, and these few parts are the same size. Okay, and the size of the quadrat depends on the types and also size of the organism and the area of the study. The bigger the area, you need more quadrat and bigger size of quadrat. Okay, so it depends on the size and also types of organism and the area of the study. So what are the data on population size that this quadrat sampling can estimate? Right, so there are three. The first one is frequency of the population. The second one is density of the population, and the third one is the coverage percentage of the population. Frequency. Frequency indicates the number of times a plant species is present within a given number of sample quadrat. And the formula is the number of quadrat containing study species divided with total number of quadrat used times 100. For example, let's say this is the area that I want to study, okay, and I want to calculate the frequency of sunflower in this area. So I put 7 quadrat, 1, 2, 3, 4, 5, 6, 7. So I have 7 quadrat used. Now I have to count the number of quadrat that containing sunflower. So I have 1, I have 2, I have 3, I have 4, and I have 5 quadrat containing sunflower. So put inside the formula. So I have 5 divided with 7 times 100. So if we count, the frequency is 71.4%. Density. Density is the average number of individuals in any species per unit area of the study. and the formula is the total number of individual species studied in all quadrants divided with the total number of quadrat used times area of a quadrat. Okay, this one I give you two examples. We look at the first example. This example, the area of study is only this small. Okay, so I want to study the density of this bunch grass species. Okay, how to calculate total number of individual species studied in all quadrants? So how many we have? 1, 2, 3, 4, 5, 6, 7. Okay, because we only want to study bunch grass species, so we have 7. Okay, divide with total number of quadrat used. Right, since the area is small, so I just use all the quadrat. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. So I have 36 altogether, number of quadrats. Area of a quadrat. Okay, let's say the area of a quadrat, meaning we take 1, let's say this is 1 centimeter square. 1 centimeter square, so the area is this one is 1 centimeter, this one is 1 centimeter, so the area is 1 centimeter square. So put it in the formula. We have 7 just now divided with 36 times 1 centimeter square. Okay, punch in your calculator and you will get 0.19 individual per centimeter square. Okay, for the bunch grass. Let's say I have a bigger area. Okay. And I want to calculate the density for this dandelion flower. The yellow one is the dandelion flower. So I put 2 quadrat, the big one. Okay, 2 quadrat. So I need to calculate the total number of the dandelion. Okay, in the quadrat. So I have 1, 2, 3, 4, 5, 6, 7, 8. I have 8 altogether dandelion. The quadrat used, okay, the big one. Okay, I have 2 quadrat used. Okay, area of a quadrat. So let's say this one quadrat, the whole one quadrat is 1 meter and 1 meter. So the area is 1 meter square. So we put it into the formula. So we have 8 divided with 2. That is the number of quadrat times the area, 1 meter square. Punch in your calculator and you get 4 dandelion per meter square. Coverage percentage. So what is coverage? Coverage is the surface of the soil or the place of study or the area of study which is covered by the shoots of the plant species. Okay, and to calculate the coverage percentage, we simply times it with 100 to get the percentage. So the formula for coverage percentage is area covered by the study species in all the quadrants divided with total number of quadrat used times area of a quadrat times 100. Again, I have 2 examples for you. The first example is to study the species of Mucor sp on a bread. Okay, so I use the whole quadrat. Okay, so first, area covered by the study species in all quadrat. To calculate the area covered, we only count the area which has more than half. Okay, meaning the Mucor species must cover more than half of the area. Then only we can count the quadrat. Okay, let us try. Okay, so we have this one, 1, 2, 3, 4, 5, 6, 7. So we have 7 here. Okay, how about down here? We have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So we have 10 here. Okay, so the area covered by Mucor sp on this question is 17. Okay, divided with total number of quadrat used 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So we have 10. 10 times 10 is 100. Okay, 100 of quadrat used times area of a quadrat. Let's say this one area is 1 cm, this one also 1 cm. So the area is 1 cm square. Put inside the formula. Just now we have 17 cm square divided with 100 quadrat times 1 cm square times 100, punch in your calculator, and you have 17% of the coverage percentage of nuclear SP on this bread. Let us try on this lichen, which is on a tree trunk. Okay, the yellow color or the brownish color is the lichen on the tree trunk. Okay, we try to count the area covered. Okay, so which one has more than half? So we have 1, we have 2, we have 3. Okay, the rest are less than half. So we have 3. Okay, let's say again this is 1 centimeter, 1 centimeter. Okay, let us count the number of quadrat. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So we have 100 quadrat altogether. Area of the quadrat is 1 centimeter square. So just now put inside the formula, we have 3 centimeter square, the area just now, divided with 100 quadrat times 1 centimeter square, that is the area of a quadrat. Punch in your calculator and you get 3% of the coverage percentage for the lichen on the tree trunk. Okay, revision questions. Okay, 2 students visited a location in Samarahan and counted the number of lichens on 3 trees at that place. They cut out the 10 cm times 10 cm square of transparent plastic to use as quadrat, which they attach at the bark of the tree trunk using adhesive tape. Then they count the area covered by the lichen in each of the quadrat, and the results is as shown. Okay, you have quadrat A 50 cm square, quadrat B you have 30 cm square, quadrat C you have 65 cm square, and quadrat D you have 45 cm square. Right. Okay, so the question, calculate the coverage percentage of lichen in all quadrats. Okay, so first we put in the formula. The formula is area covered by study species in all the quadrats, meaning, okay, we have to count 50 plus 30 plus 65 plus 45 because it is in all the quadrats. Divided with the total number of quadrats used, how many quadrats we have? A, B, C, D, 4 quadrats. Times area of a quadrat. Area of a quadrat, just now I mentioned in the question, time 10 times 10. 10 times 10 meaning, okay, it is 100 centimeter square. Okay, so put in the formula. So if we count, okay, 50 plus 30 plus 65 plus 45, we have 190 centimeter square, divided with the number of quadrat used, that is 4, times the area of a quadrat, that will be 10 times 10, that is 100 centimeter square. Okay, and then times 100 because it is the coverage percentage. Punch into your calculator and you get 47.5%. Okay, you want more exercise? Okay, you can find the link below this video. Okay, click on the link. There are more exercise on this topic. You can try it. Don't forget to subscribe my channel to get more update on a new video. Till then, bye-bye.